This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1117, recorded on May 24th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. One of those perfect days out there. It's 83 degrees Fahrenheit. The humidity is something like 52%. And it's a gorgeous day. There's no high winds or anything. Should be outside.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 81 and partly cloudy, wispy clouds. Too bad it's not 82 because then it would be 28 Celsius, but it's pretty close to that.
So the benchmarks are 20, right? So which is 68. 28, which is 82. Are there any others?
16 is 61.
Okay, 60 to 61.
And 28 is 82.
82. Perfect. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Long time no see.
Yeah.
It's
Good.
been
Welcome back.
At least 5 weeks or something like that. And we have imperfect weather here. It is 93 degrees and kind of sticky and overcast and generally sort of yuck.
Well, welcome back. Good to have you again.
Thank you.
Good
to be here.
We missed you.
Well, I missed you guys too. You people too. Guys, well, we did that.
Whatever.
You go back to episode whatever. Right, we'll translate guys for you. Okay,
That's right.
Yes,
That's right.
I missed you too. It's good to be back.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And here it's 84 Fahrenheit, 29 C. Also missed the 82, 28. Humidity's 22%. It's gorgeous. Lovely, lovely day. I went out for a walk to a local park earlier. Just beautiful weather today.
And from Madison, New Jersey, Brianne Barker.
Hi, great to be here. And it is just as beautiful here as others have mentioned where they are. It's 84 Fahrenheit and wonderfully sunny and great weather for a fun weekend.
Brianne, you have a different position. It looks good.
Thank you. Yes, I rearranged parts of my office and think that this seems a little better.
Yeah, it looks more casual. Looks like you're not sitting in front of a big screen, the books in
I'm sitting
the
a
back
little
there.
further from the big screen.
It's great.
And you're in a
It's
different setting too, Vincent.
I'm in a little town in Italy called Terriciola, which is in Tuscany, where, by the way, it's cooling down now. It gets very cool at night. It is currently 17 Celsius, just went down a degree, and partly cloudy.
Yeah, what time is it for you?
It is 9:06 p.m. So we're 6 hours after New York City. So thank you, Dixon, Brianne, and Alan, for taking care of last week's episode.
An extreme pleasure.
No problem. It was very fun.
Fascinating.
Feel free to take 2 or 3 weeks off.
Well, no.
Yeah, I was going to say, that kept us on our toes.
No. Yeah, we appreciate you being here.
I will be there. So next Friday, I will be in a small town 3 hours north of Helsinki for a small European coronavirus meeting. So Friday, 7 hours after New York's in that part of the world. So it'll be 10 p.m. I can do that. There's no meeting at 10 p.m. The following week, I may or may not be there. We'll see. Depends how things go. But I should be mostly here for the rest of the summer. So, but anyway, I know now that I can count on you guys to do a great show. Daniel loved your banter last week, so good job.
Oh,
That's
good.
good. That's good. Yeah, we had
All
fun.
right, let's see here. Now, I only have one stinking little laptop screen. I don't have like the 3 screens that I
You're
usually
going to need to use the
have.
workspaces and page between them, right?
Yeah. If you like what we do on these podcasts, we'd love to have your financial support. Microbe TV is a 501c3 nonprofit, which means in the U.S., your deductions are federal tax deductible. So you can go to microbe.tv/contribute to learn more. Kathy, what do you have for us?
Well, the first one is a PSA that I didn't put in. It's about the International HBV meeting. Do you know about that, Vincent? Do you want to talk about that?
Okay. Yes, I think Karen put that in. And that is because we're doing a TWiV there. This is the International Hepatitis B virus meeting. Very timely because we're going to do a Hep B paper today. It's going to take place September 11th through 15 in Chicago, which is in the state of Illinois in the country of U.S. And you can go to hbvmeeting.org for information, deadlines, registration, abstract submission. And we will do a TWiV there with 2 of the meeting participants. So that should be a lot of fun. Michelson Prizes, that's not you, is it, Kathy?
I did put it in,
there.
but you
Yeah,
can
go ahead.
you can do it.
You
Well,
want me to?
sure, you do it.
I think we already mentioned these. The Michelson Prizes, Next Generation Grants, annual amounts of $150,000 to support early career investigators who are working to significantly advance human immunology and vaccine and immunotherapy research for major global diseases. These prizes are awarded by the Michelson Medical Research Foundation and the Human Immunome Project. Applications are being accepted from April 1st to June 9th, very soon. And we'll put a link in the show notes if you want to
And my
apply.
apologies to the Michelson Medical Research Foundation. I think I pronounced them Mitchelson last time, so my mistake.
I'm not sure we know how to pronounce it. But...
In that case, apologies to whichever pronunciation is wrong. Yes.
Kathy.
All right, I have some news about ASV 2024, which is coming up in just about a month. A month from today, June 24th to 28th is when the meeting is. So the special events and workshops are in the program PDF when it gets sent out and the mobile app. But meanwhile, you can just see it on the meeting website under the program tab, listed just below the satellite symposia section. And so there are a number of these that you may want to check out. One of them was mentioned last week, but I want to call your attention to one that happens on the very first day of the meeting, just before the keynote, sort of concurrent with the welcome reception. It's a DEI panel on allyship in virology. And this year it's going to be with Black Microbiologists Association. So it's supporting and advocating for Black virologists. So that's Monday at 4:00. And then there are others that we'll continue to describe. You heard last week about the career one, Think Like an Actor, Speak Like a Pro. There's an education one, there's a communications one, and there's an assistant professor boot camp. And then the other thing I wanted to start reminding you again, if you attended ASV in Athens or Madison, so last year or the year before that, you should start looking for your name badge pouch. We are trying to be eco-conscious.
If you look straight forward, your flight attendant is holding up an example of the name badge.
Yes, here's another one.
Yes, there's another one. Okay.
And so the idea is that, as you can see, this part is generic. It doesn't have a year on it. And then this part you will get filled in when you get to the meeting. And so if you can bring these back year after year, you don't collect them in your office or in your wastebasket. And it makes us more sustainable.
And for those of you who are listening and not watching the video, that's a black pouch that says American Society for Virology on it with a clear window in the front.
Right. That's it.
Right. Thank you, Kathy. So we have one item in our news, and this was sent by Kathy. And this is a news release at the NIAID, National Institute of Allergy and Infectious Diseases site. It's called High H5N1 Influenza Levels Found in Mice Given Raw Milk from Infected Dairy Cows. And I would just like to fix the title to read High H5N1 Influenza Virus Levels. Sorry to be pedantic, but you don't have high levels of disease in mice given raw milk. Anyway,
actually, it's probably I haven't actually looked at this, but I'm guessing it's high H5N1 influenza virus RNA levels.
Yes, I suspect so.
That's right. Okay, so this is about raw milk, and the cows, as we discussed with Richard Webby a couple of weeks ago. Many dairy cows have sustained infections in their mammary glands with avian H5N1 influenza virus. It's shed in milk. And they say that 52 cattle herds in the U.S. across 9 states have been affected, and there are 2 human infections in dairy workers with conjunctivitis. Okay, so what they did is they said, is there a problem with raw milk? Because some people like to drink raw milk, which means it's not pasteurized or heated to destroy bacteria and viruses. So they took raw milk from these infected cows. This happened at University of Wisconsin-Madison and Texas A&M, and they fed droplets of raw milk from the infected cattle to 5 mice. And these mice demonstrated signs of illness, including lethargy on day 1, and were euthanized on day 4 to look at virus in organs, and they discovered high level of virus. Now, this is PCR, I presume, right? So,
Well, I don't
I'm
know,
looking at the
actually.
paper right now, trying to figure that out.
okay,
Yeah, the
I
paper's in the New England Journal of Medicine.
right.
I didn't go to that, but yeah.
No, it looks like it's a plaque assay.
Yeah, I wouldn't be surprised.
Nice.
Good.
Yeah.
TCID 50,
Yep. So I
Excellent.
take back my facetious correction of the title.
as well as looking at some eggs.
So they say
Wow, eggs. Cool.
Yeah.
high levels of
Those are real
virus
virology assays.
Yeah.
in nasal passages, trachea, and lungs, moderate to low virus in other organs, consistent with H5N1 infections in other mammals. They also did an interesting set of experiments to try and inactivate the viral infectivity in raw milk from dairy cows. So they had 4 milk samples with confirmed H5N1, and they tested heating at 63 degrees for 5, 10, 20, and 30 minutes, or 72 degrees, these are Celsius, for 5, 10, 15, 20, or 30 seconds. At 63, each of those time intervals destroyed infectivity. At 72, virus levels were diminished, but not completely inactivated after 15 and 20 seconds, but the others, 30 seconds did. Of course, this is a lab study, so it may not happen in the real world. But pasteurization, what's the time for pasteurization? Does anyone know offhand?
It's quick.
I think there are a couple of different
Yeah, there are
protocols for it,
different
and
ways
there's
to do it.
ultra pasteurization, and I think you can do it for a short period at a higher temperature or a longer period at a slightly lower temperature.
Yes.
yeah.
But I don't remember what the specs are exactly.
It also seems like it would be important at what volume of
Yeah, that's
container you have the milk in, and
Right here.
Yeah.
scale up from a pilot
Right.
experiment.
Right. And at industrial scale, it's done so that the entire volume of milk reaches the pasteurization temperature either by heating the whole container for a long enough period of time or by piping it through a pipe that heats the small amount rapidly and then cools it back down.
In most processing plants, chilled raw milk is heated by passing it between heated stainless steel
Yeah.
plates until it
Correct.
reaches 161 degrees Fahrenheit, and then held
161, right.
and then held at that temperature for at least 15 seconds,
Okay, so
according
that's
to the usdairy.com.
that's this probably 72 degrees Celsius. Yeah, 161.6 is 72 Celsius.
Yeah. All right. So the bottom line, folks, is if you're drinking unpasteurized milk, which came from an infected cow, it's going to have infectious virus in it. And how did they infect the mice? They dripped it in their nose. So there's a risk of getting infected if you're drinking unpasteurized milk.
They
Wouldn't you say
fed
that's the bottom
droplets of
line?
raw
Yeah,
milk.
they were
fed
fed
them.
droplets at the back of the
Fed.
throat with a pipette.
Fed.
Interesting.
So it wasn't
that
into the
makes an
nose, it
aerosol.
was
That
yeah.
makes an aerosol, right? As you
Mhm.
swallow,
Mhm.
so
Us. Was-
you can get
for us.
infected. So might want to rethink unpasteurized milk.
Although, as I commented last week, we roll our eyes at people drinking raw milk, but a lot of people, including some probably on this show, eat raw oysters. Some people eat raw beef. So
I don't eat raw oysters anymore.
I don't
Yeah,
eat raw
I
oysters.
agree with you.
My wife does. My daughter does.
Yeah,
Most of
it's
my
okay.
family. I'm a terrible Marylander. I don't eat raw oysters, but most of my family does. So
Okay, we'll put a link to that in the original paper in the show notes. For our snippet, we have a very interesting paper from Nature Microbiology, Dynamics of Measles Immunity from Birth and Following Vaccination, from Wang, O'Driscoll, Wang, Zhao, Salji, and Yu.
And Salji and you are co-senior authors.
No, I'm not.
Not you. You. Who you
Yes,
calling a you? And this group is from Fudan University in Shanghai and University of Cambridge, UK.
Right. So some of the questions that they want to address in this paper. First, how long does immunity to measles last conferred by vaccination? And in this paper, they're looking specifically at antibodies.
Mhm.
And do maternally derived antibodies, cause reduced vaccine responses? Because as you know, if a mother has antibodies to measles, they will be passed over the placenta to the developing fetus. And there's some evidence that interferes with the take of measles vaccination.
Right.
So,
And I think that one thing that I really found sort of important here is that we know that there are maternal antibodies against measles, and we sort of imagine that everybody's responses are just entirely the same. And so once upon a time, someone, because they didn't have the ability to do these high resolution immunological studies on huge numbers of people, said, okay, we're going to make the measles vaccine schedule where we're going to vaccinate kids at a particular time because we assume that the maternal antibodies will be gone and there won't be any more of a problem from those maternal antibodies. And so there weren't necessarily a lot of measurements. Makes sense because people don't necessarily want to give their babies samples for measurements, but there was just this idea of if you wait to a certain amount of time, and as I learned in this paper, the amount of time is different in different countries, that you just say they're fine, and assume that the maternal antibodies are gone, and assume that you're going to be all set, and that it's the same in everybody.
Yeah,
They're never really gone though, are they? They're just are cut in half and then cut in
they
half
wane
and then
over
cut
time.
in half.
Yeah.
But they wane, but they don't go away.
I want to sort of hammer this in a little bit for people who haven't thought about this before or much. So the moms have antibody in their blood, okay, that can cross the placenta, and babies are born with it.
Yep.
And that can then, if you try and vaccinate those kids, theoretically, those antibodies can react with the vaccine and make it so that it doesn't work as well.
Exactly.
I guess the point I want to make is that the antibodies coming from the mom are like a passive immunization. That is, it's just the antibodies. The what the kids get from the mom does not allow them to make new antibodies from the mom. It's just protein that's around and then goes away. And they have to then be immunized so that they have their own cells that make antibody. All right.
Right. So you should say that it's only IgG and not IgM that the mother transfers across.
Right. And it's only antibodies. It's not the ability to produce antibodies.
Exactly.
Right.
Or any other immune response.
And this timing thing is kind of a guessing game. So, well, I don't know if this tries to get out of the guessing realm, but there's a good news, bad news situation, right? You want to wait long enough so that the maternal antibodies won't compromise the vaccine. But as you wait, the kid's immunity from
they become more
the mother
susceptible.
Decreases, they become more susceptible. So if you wait too long,
right,
you're exposing the kid to risk. All right. So what's the sweet spot?
I think in the U.S. it may be 1 year.
In the U.S. it's 1 year.
Yeah.
Many other countries do that. China, it's a little earlier, as they talk about in this paper. And one reason they're looking at this is because the measles vaccine is not 100% for everybody throughout life. And they point out that in China in 2013 to 2019, a third of the measles cases were in people who'd been vaccinated. So that represents a vaccine failure, and it would be nice to try to figure out why that's happening. And to get to that, you go back to the beginning and say, well, okay, when we gave the vaccine, did it take, or were the maternal antibodies a problem, or was something else a problem? And to Brianne's point, how different is this from one individual to the next?
I would try to make a parallel to the idea that having the vaccine and what they are calling a failure might still allow protection from
Yes.
disease. And it's
Mhm.
something that we've never really looked at much for some of these other things. And that maybe these people who got the measles got a less severe case of the disease, but none of that's even addressed here. So
Right.
it gets to this whole idea of, I can't think of the term that people object to when we talk about the breakthrough
Yes.
infections. That is it really that the vaccine failed, or did it do what it was supposed to and gave you a less severe disease?
Right.
So the trial you'd have to do to answer these questions are pretty difficult to do. Like you'd need to have baseline serum or when the child is born and late, and it's often difficult to get. But they take advantage of a immunization program in China, and they apply statistical methods from which they can predict what's in the baby based on maternal antibody titers. It's very cool, as you will see.
Yeah.
Yeah.
And it's this China study. Basically, they did nationwide supplemental immunization with measles from 2004 to 2010, and these are kids 8 months to 14 years of age, no matter whether they had been immunized or not. And so they have data from cohorts of children there. It's in Hunan, China, which have been published before, and they basically are reanalyzing them in this paper.
And two cool things about this cohort. First of all, it's a large group that they were able to get follow-up data on. And they have infants and they have kids up to 12 or 14 years old. Or no, 9, sorry, in this, when they
Yeah.
started the study. 1,500 participants. And the other cool thing is because of these supplemental immunizations that Vincent mentioned, some of these kids had up to 5 vaccine doses. The normal course is 2. 2 or 3?
Yeah, 2.
2.
I think
2.
it's 2. So, so they have this range of doses, and they have this large population, and they've got infants and kids at different ages, and they can feed all of that into this model.
Yeah, and they also have antibody levels and samples from moms,
Yes.
Yep.
so that they can actually look at what was going on in terms of moms' antibody levels. And we hadn't really mentioned, but one reason why all of this is important is because there have been increasing numbers of measles cases. Unfortunately, there are decreased percentages of people who are getting measles vaccination, and so there's also some questions of, do we need to make any changes to what we're doing with regard to measles vaccinations? And this makes some nice, really kind of studies this and lets us make evidence-based answers.
Yeah, and they
And
looked
since
at.
oh, sorry.
That's okay. Go ahead.
Well, since I was going to say, since it includes kids who had up to 5 doses, it addresses one of the obvious things you could do, which is, do we need more boosters? So that's the
Yep.
experiment's
Yep.
kind of been done, and now they're looking at
Yep.
the data.
So they also looked at cesarean delivered children.
Yes.
Mhm.
Yep.
And that's an incredibly interesting control. And so you could vaccinate those children almost immediately without worrying about whether the antibodies would interfere with the vaccine or not,
Well,
because there wouldn't be
No,
any.
no,
Yeah, there
they've
should be.
Or
Should be.
would
There
there?
are.
No, I'm
Yeah, there
sorry.
would be. There would be.
Okay, that's
Because
okay.
the antibodies pass during
Erase.
development. Yeah.
Right.
No,
So,
sorry.
well, let's look at the data. So the measles in the mother, the maternal measles antibodies in cord blood, right? That's how they measure it. At birth, had a mean concentration 6.42, right? Log concentration, but more importantly, they see a high correlation between the titer in the cord blood and maternal. So that means you can use cord blood, right, and not have to have separate blood draw from the mothers. They found differences in concentrations based on the sex of the infant. So cord blood antibody titers were about 1.03 times higher in male versus female infants. But these different rises by 6-7 months of age before you get the first dose of measles vaccine. But after that first dose, this trend is reversed. Females have 1.04 times the concentrations of antibodies in male infants. And that is a sex-specific difference that we've talked about before, which is well known. That there's
Yeah,
a
and it's
strong
also
fact.
kind of cool because oftentimes when we look at sex-specific differences in immune responses, we're looking in adults,
Yeah.
where we know that there are going to be a lot of hormonal differences, and we wouldn't expect the same degree of hormonal difference in these infants.
Yeah, and this is following the first measles immunization, right? So here they're given
Right.
a chance to have an immune response, and right away, the female children have a 1.04 higher levels of antibodies. They also looked at the decay of antibodies. So maternal antibody has a half-life of 181 days, and which they say is consistent with other studies. So their data are good.
Yeah, remember that number, Dixon, because that'll be
181 days.
compared.
Yes, it's important. Okay, so then they build models with these data to try and understand antibody concentrations following vaccination. And so they first look at what model to use, and they decide that exponential decay model is good. And they find that there is a peak log concentration increase of 4.73 after that first measles vaccine, and there's big variation across infants. Right, the range is huge.
Yeah.
And then there's a slower rate of decay for the antibody after immunization with measles compared to the decay of the maternally derived antibodies.
Which makes sense because the immunization is stimulating the kid to produce their own
Exactly right,
antibodies, so it's
right.
more sustainable, whereas the mom's antibodies are just there. Whatever initial amount you were born with is then decaying over the 181 days.
It's a pretty impressive difference, though. It's 181 days versus 16.1 years.
Yes.
16.1 years half-life, yes, following
Yeah.
vaccination. That's the first dose, right?
Yep. And by the way, both the papers we're talking about today are unfortunately paywalled, but if you can get them and you look at these figures, you see these phenomena very clearly in the graphs.
What is interesting also is that there are responders. 324 out of 350 infants were responders, right? They
to
made
the first
antibodies
vaccine.
to the first vaccine,
immunization.
Mm-hmm.
and then 26 out of 350 were non-responders. They didn't make a good antibody response. So very clear bimodal distribution. All right, so why?
And even within the responders, there's this wide variation.
Yes.
There's a lot of diversity.
All right, so what correlates with this difference, right, after the first immunization? So there's a strong association between maternal antibody before the vaccine and peak responses to the vaccine. Basically, with each maternal log concentration increase, you get a 1.44 reduction in the vaccine-induced concentrations of antibody. So the higher the maternal titers are, the worse the baby responds to that first immunization.
Which is consistent with the model of immunological damping, right?
It is. And it's also really interesting because they find that they used a model based on maternal antibody levels and a model based on just age, which is what we currently do to say when is the right time to get your vaccine dose. And it turned out that if they used a model based on mom's antibody levels, you got a much better response if you kind of picked timing. the models predicted that was going to actually predict your antibody response much better than your age was.
So wait a minute. I went dark for a minute.
Okay, so they
So no. Back.
okay,
The
yay.
the non-responders. Did I hear Alan say that correlates with the notion of the dampening from the mother's antibody?
The
Or is it,
degree of response you get is dependent on the amount of antibody,
Yeah.
on the amount of maternal antibody present
Among
in
the
the
inversely-
responders.
Among the
Inversely
responders, right?
correlated. So-
Inversely
Okay.
correlated, yes.
Yeah, because you get a
Okay.
big range of response in the responders.
Right.
and
In
that
the
correlates.
non-responders, they're non-responding.
Okay.
And
Yeah.
you've got
Okay.
26
We'll find out
kids
what
who
So you got a
goes on
certain
Yeah, we'll
you got a
find out
certain
more
number of
about that.
people who, regardless of the circumstances, just don't respond
Yeah.
to the first vaccine. Okay, that's what I
Right.
wanted to
And of the
clarify.
ones who respond, there's an inverse correlation between
Fine.
maternal antibody concentration and level of response.
Great. Okay. Good.
Also interesting is that the infants born to primiparous women, so it's their first or their only child, they had 0.23 higher peak log concentrations compared with infants of women who had many children, multiple, more than one. And then again, female infants had 0.22 higher peak log concentrations compared with male infants. But infant age at vaccination, vaccine type were not associated with this variation in the responders. But maternal antibody concentrations explain 56% of the variance in this response to the first vaccination in the responders, right? 56% of them.
I think 56% is huge.
Yeah.
Yes.
When you're explaining variation among things, I
Yeah,
just remember this
sure.
from the genetics of susceptibility. If you had that kind of contribution, it would be huge. Although
Right.
to the non-cognoscenti, it's at like 56%. That's
Right.
not
Right.
accounting for much.
In an outbred human population,
Yeah.
that's a really
That's a big
number.
deal. Yeah, you don't see effect sizes like this very often.
So here's where it gets interesting. They want to develop models where they could use the measurements of antibodies in the mother to figure out what's in the baby, right? So they don't use any infant antibody measurements. They could estimate the peak antibody concentrations after measles vaccination 1 based on simply what is in the mother, because there's a huge correlation between maternal antibody levels and what's in the infants. And they say this is cool because we can do personalized vaccination, right? The age of that vaccination can be guided by concentrations of antibodies in the mother to maximize the protection of the infant.
And you don't even have to take a sample from the baby.
You don't
Right.
have
You
to
could
take
imagine
a
some
sample,
people
right?
would be really upset about.
Right.
And
You
you
can
just-
take cord blood and you
cord
were going to be
blood.
throwing the umbilical cord away anyway. So
Exactly. So it's
free
it's
sample.
good for everybody. You don't have
Yep.
to stick the mother again. You just take cord blood.
So you could actually, as a routine part of a delivery, measure the antibody levels in the cord blood
Yep.
and do the appropriate calculation and say, this kid ought to have their first dose of vaccine at X months.
Yes.
That's right. All right. Now they want to know why some of these babies do not respond to that first vaccination. So they look at all the variables that would distinguish the children. And the only thing that's significantly associated with failure to respond to the vaccine is the mode of delivery, where if you were born by cesarean section, you had a 2.2 to 2.6 odds of primary vaccine failure compared to babies born by vaginal delivery. And so the numbers of the infants born via cesarean, so there were 133 in this study, 16 or 12% failed to mount an immune response to the first vaccine, whereas 217 infants born by vaginal delivery, 10 of those, or 5%, had vaccine failure. And so they say this has been found for responses to other vaccines as well, this mode of delivery. And you get a different microbiome when you're delivered by C-section. And so they think
True.
that may be influencing the immune response, which I think is very, very cool, right? Because we
Mhm.
know
Yeah.
for years now that the baby microbiome is very different with a C-section, and now we're seeing it's even affecting vaccine responses.
What about the second vaccination?
That's the next part of the paper.
That's the next. Yes.
Bingo.
Excellent question.
All right, so they studied that,
Okay.
and they find that the infants who failed to respond to the first vaccine were able to respond after the second one,
Right.
right?
And
Which
why
is
do you think
great.
that might be true?
I don't
Well,
know.
following on the microbiome thing,
Yeah,
they've
following
had
up on the microbiome, you see, now
if
you're going
that's
to
the
give that
deal,
infant a chance to catch up and acquire the mother's microbiome from nursing and from being just close to the mother. So they may have recovered their original microbiome by simply close association. I don't know what they give the reason for, but that would be a logical explanation.
Well, they suggested it's microbiome related. Yeah. So
Yeah, the microbiome is
Right.
maturing, and the
Right,
second
right,
dose
right,
is
right,
coming
right,
later, and so
Right. Cool.
all right. So then, how about an age specific when should we vaccinate kids? Basically, right? So they have models where they model in 3 vaccination schedules for the first and second doses of measles vaccine. So 6 and 15 months, 8 and 18 months, and 12 and 24 months. And these,
And I'll just point out that 8 and 18 is basically what they're doing in China, according to this paper.
right?
12 and longer than 12 is what we do in the US, though it's more like 12 and 48 instead of 12 and 24.
we do 12 and a long time.
Yeah.
It's like 4 years or
Yeah.
something like that.
So we've been talking about log titers of antibodies, right? So you may be wondering, well, what do you actually need to protect a child from disease, right? So they say the threshold of measles immune protection is 120 MIU per mil. And so they can use that number to estimate based on the output of their model where they plug in at these different timings, the first and second vaccine. And that, of course, is based on the data they've just collected, right? They estimate that 0% of infants are still protected by maternal antibodies when you get the first dose at 8 months, compared with 4% or 0% at 6 or 12 months, right? So basically, waiting 6 months, you lose all protection against measles disease. 0% are protected. Then if you do MCV1 at 8 months, 100% of the population is immune following the vaccine with the right antibody concentrations. And it's comparable with 99% protected if it's given after 6 months, and 100% protected if you give it at 12 months. So they decide, based on this and more that I haven't described, that 12 and 24 month schedule gives you higher and longer lasting antibody concentrations, with a slight risk of kids getting measles in that interval, right? So in those first 12 months of life, your maternal antibodies are going down. You could get measles, but you get a better
there's
response.
an extra
if you
4
wait.
months, 4 months of vulnerability in
Yeah.
that schedule.
Yeah, but like you have a longer lifetime protection, and you have a higher overall protection if you get that 12 and 24.
Right.
So that's the study. It's very cool. So first of all, you can
So-
predict what the child's antibody levels are going to be after vaccination from the maternal levels. We know that cesarean section birth is not great for responding to that first vaccine, although you do respond after the second vaccine, so you should get 2 doses for sure. And that the best timing is 12 and 24 months. And by the way, something we didn't touch, doses beyond the second give you marginal increases in antibodies, so they're not really needed. And they say that something is very
the kids who
interesting.
got 5 shots are not better off.
Right, but some people are using measles vectored vaccines for other vaccines, right? And this has implications for that, right? Because after those first 2 measles doses, you're not going to respond very well to a measles vectored vaccine. And that was observed in at least one measles vectored SARS-CoV-2 vaccine, which failed in human trials, probably because they had high antibody levels after vaccination. So,
Or at least high immune responses.
Yeah, high something.
Yes, they even comment in the paper, we do not consider cell-mediated immune responses to vaccination, which are often longer lived than antibody-mediated immunity.
right.
So at least they mentioned it.
So would there be a downside other than an extra stick of vaccinating children fairly early to get to, if you weren't going to do the personalized thing, to get the kids whose maternal antibody had waned significantly during that early period, and then vaccinating everybody again at, say, 12 months. So like 4 and 12 months or something like that. And then the third immunization.
Well, they didn't model 4
Well,
months. It's a good question, but we know that there's still maternal antibody left at 4 months, right? And most kids, and so that could not have a good outcome, I would guess.
Yeah, I think the marginal benefit of it is probably not going to justify the cost and the extra stick.
So it'd be interesting to see superimposed on this a measles incidence correlated with age. Okay, so how big a deal is this? How many kids in the first 12 months of life get measles?
especially if we think about this in maybe different populations where there are different incidences of measles
Yep.
in general, at what point does
Mm.
that risk become enough to justify that additional vaccine dose?
Right.
Yeah, I'm looking at that figure 3B where the green curve, I'm wondering maybe if it was at 8 months, and then if you look at how it doesn't seem to really wane much differently from the 12-month vaccination, maybe that would be the way to go. But I'm also, as Rich was saying, it would be nice to know something about the measles incidence, and then also thinking about the fact that measles causes you to lose immunity to other things.
Mm.
Maybe by the time you're only 8 months old, you haven't got as much immunity to lose, but still, or how often do infants progress to SSPE? I don't know that kind of information, but it's something to consider, not just from the standpoint of, protection from measles, but protection from all those sequelae of measles.
So they do some numbers based on China. They say basically 1.15 million children aged 0 to 4 years would be susceptible to getting measles in that first year if you did 12 months for the first vaccination. 1.15 million. So it's a lot of kids. But it would depend on, they're likely to be scattered, right? They're not going to all be in one place.
This is where
So-
herd immunity now
Yeah. So
steps in.
Right.
Okay.
if you have a highly vaccinated population against measles, right, 95%, less of a risk, right? But as you see,
Very
now,
interesting. It really, this whole thing really shines a light on the whole discussion that we alluded to several weeks ago. Okay, and puts numbers on it. It's very interesting and cool data, cool experiments.
Yeah,
Yeah.
a lot of modeling. And I like the predictive value, as Rich was saying, if you could get that information early on and then say, okay, this kid needs their dose of measles vaccine at this age, this number of months.
Yeah, I'm picturing the nurse and the obstetrics looking at a table and saying, "Okay, you should get your measles vaccine on this schedule."
Right, exactly.
Right.
they also
I'm
say
also
that
curious about the role of maternal antibody with regards to mother's milk and breastfeeding, as to whether or not that could prolong the amount of mother's maternal antibodies in the child.
I think that they mention in a couple of places where breastfeeding status was not associated with vaccine failure of the first dose.
Uh-huh.
but that's really the only place where they mention much about it.
Yeah.
Mm.
So this is all about antibodies, right?
Yep. Yep.
We have said nothing about T cells or cell-mediated immunity. Now,
Nope.
you're not going to get any of that from the mom, right?
You're not, no.
You're just going to get antibodies.
Right,
But when you do the vaccine, you're going to get cell-mediated immunity as well, right?
exactly.
Which I don't know how big a role that plays in measles, but I can't imagine that it's zero. And I don't know how to factor that into this discussion at all.
I also wanted to know whether or not, even though the non-responders didn't respond to the first vaccine dose, but did respond to the second vaccine dose, was the second response commiserate with whether they had been exposed before to another vaccine dose? Or was it like a first vaccine dose rather than, in other words, did they produce IgM antibody first rather than IgG antibody first?
Mm. Yeah, they don't mention
That
that.
they didn't comment on.
They didn't
They
talk
did-
about
yeah.
Because there might have been a
isotype.
small response that wasn't measurable, but nonetheless they did respond.
Yeah,
And
sure.
then that would have preempted the fact that the
Yeah,
type
they
of
only talk
antibody.
about. I think they only talk about antibody levels, and in the non-responders, the level change on the second dose was the same as the level change you saw in the responders on the first dose.
Mm.
They,
The
well,
responders on the second dose didn't really respond much to, or on the first dose didn't really respond much to the second dose.
I
which
see what
makes
you mean.
sense.
So it would have been about the same, I see.
yeah, so
Whether
they say post measles dose 2, we find non-differentiable peak levels between the responders and non-responders. So basically, at dose 2, the two groups don't look different.
Right,
Right.
but they don't look at class switching,
No.
They don't look at classes.
or at least they don't talk about it.
That's right.
We are nerding out here. folks.
Yes.
You
This
got
is
it.
great. This is
That's
great.
right. That's what we do. That's what we do here on TWiV.
Well, it's a cool paper.
Yeah,
It
it
is.
is
It's
very,
very cool.
Yeah.
Very cool.
Yeah.
All right. Our next
Speaking
paper.
of a nerd fest,
This one is nerdier because
yeah.
first of all, I
Oh
think
boy, is it?
hepatitis B virus is a very cool virus.
It's
And
a
this
nerdy
paper-
virus to start
This
with.
paper is all molecular biology, which
Very
is what
cool.
we all used to do, and which is very hard to find anymore. And so
It's
that's
even
what
got
this is.
northerns in it. Holy cow.
This is a very cool paper. It is published.
They ran
in
protein gels. My eyes were watering. Yeah.
Cell. Deep mutational scanning of hepatitis B virus reveals a mechanism for cis-preferential reverse transcription. And so we have two first authors, Yingpu Yu and Maximilian Cass, and two corresponding authors, Charlie Rice and William Schneider. So this comes from the Rockefeller University, Heidelberg University, Harvard Medical School, Weill Cornell Medical College,
And the Broad Institute.
the Broad Institute of MIT and Harvard.
To name a few other small places.
Yes.
Hepatitis B virus is a DNA virus with an envelope. It chronically infects nearly 300 million people worldwide, and we're worried about it because chronic infection can lead to liver cirrhosis and hepatocellular carcinoma. 1 million deaths a year from that. We do have a vaccine, we do have antivirals, but it's very difficult to get a cure. So you could treat someone, but the DNA, the viral DNA is always there, kind of like HIV, right?
Yeah.
You can never stop
And
treating.
HBV is it's a DNA virus that thinks it's a retrovirus.
That's right.
Right. So
This is why it's so nerdy.
yeah, I didn't look it up, but it wasn't that long ago that we had an HBV show where Rich gave the basic information about.
Yeah, and I
HBV.
think I probably made some mistakes when I did that. I think I had the well, I don't even want to say what the mistakes were, but we ought to do it again briefly to make sure it's right, because the molecular process for nucleic acid synthesis and encapsulation is important in understanding a paper.
Well,
It is
Rich,
the
if you get
core
it
of this
wrong, if you
paper,
get it wrong
yes.
this time, I will insist that you do it right the next time.
Also, I enjoy. I liked it's the core of the paper, Alan.
Yes,
That
yes.
was good.
Ooh. Ooh. So, Rich, you want to try it again?
Okay, So the virus particle itself has a proteinaceous core, which is important, and it's surrounded by an envelope with some envelope proteins in it. Inside the core, and those are S proteins, surface proteins. Inside the core is a genome that is partially double-stranded DNA. That means it's circular. So it's a circle where one strand is complete, the other strand is not. And as I recall, it's not actually a covalent circle, right?
Nope.
I think that's right.
It's kind of hooked together by the hybrid formation.
It's got a gap, single-stranded
Yeah.
DNA, and it's got a
Yeah.
piece of RNA attached to it.
Right.
So it's actually called relaxed circular or rcDNA or rich condit DNA. That's how you can remember it.
So that particle is taken up by cells, and the partially double-stranded genome is delivered to the nucleus of the cells. Oh, by the way, packaged along with this is a reverse transcriptase. Okay, the particle is taken up by the cells. The double-stranded DNA, relaxed circular DNA, makes it to the nucleus. Cellular enzymes complete that so that now we have totally double-stranded DNA. Then cellular RNA polymerase II, which ordinarily makes messenger RNA, makes a bunch of RNAs off of this thing, okay? That are messenger RNAs and also a full-length RNA that serves both as message and as what they call pre-genome RNA. Those RNAs are exported to the cytoplasm where they're translated. One of those translation products is the reverse transcriptase. The reverse, and of course, also the core proteins and the surface proteins. The core proteins assemble into the core particle and encapsulate the RNA along with the reverse transcriptase. All right. And after that is accomplished, inside that particle, the reverse transcriptase then reverse transcribes the pregenomic RNA to yield the partially double-stranded genomic RNA. It acquires the envelope and the envelope proteins and gets out.
Yep.
Cool?
And
are we
very
good?
cool. That's
the
good.
other important thing to note is that this is a very compact genome with open reading frames that overlap. So you have the polymerase gene in particular, which we're going to be talking about a lot, and the core gene. These are fairly large. And the polymerase gene, I think all the other genes overlap the Pol gene at least a little bit. Is that right?
I believe so.
Mm-hmm.
So there's a lot of overlap there, which makes it very difficult to probe, do mutagenesis and see what you can change and what you can't, and what's essential to the virus at different points. Because if you change a base and the virus doesn't replicate as a result, you don't know which protein you just messed up because they're overlapping reading frames.
And I want to try at least to introduce the concept of cis and trans with this paper as well.
Can I point out one thing beforehand?
Yep.
So just for the less aficionados or those who sort of know less about the basic virology, Rich described this process of replication of these viruses really well. And one thing that you want to be aware of is that this reverse transcriptase enzyme needs to reverse transcribe the viral RNA and not some random piece of RNA in the cell.
All the other pieces of RNA floating around in the cell.
It would be really bad if it picked the actin RNA and made DNA of that. That would be useless for the virus, sort of an evolutionary dead end. And there's also this problem of, again, sort of the encapsulation and making sure that you put the correct or package the correct nucleic acid into that core. And so this polymerase needs to be able to really specifically get to the RNA of interest.
So what's the molecular basis for that?
That's what the
That's
paper's
what
about.
the
That's what the
paper is
paper's
we're going to
about.
look at. So
Okay.
they have to.
But I think that the cis and trans point that Rich was going to make is also super important.
Yeah.
Well, if I can do it correctly, but you guys jump in if I screw this
But
up. So
before you do that, I just wanted to say that I followed along this figure in
yes,
the textbook.
Oh my
And
goodness.
it's really good. 10.24 in
Kathy,
the
you're sucking up.
5th
You're sucking
edition.
up. I'm sorry to say
I
this,
just
but you're
want to
sucking
say,
up.
for those people who have the textbook, and there are some out there who are not virologists, I know, you could look at that. It's a great figure.
Right, Rich,
So-
go ahead.
as I understand it, it's a good question is, how does the RNA know to get into the core? And my understanding is that probably the most important component of that is a signal, a structure in the RNA itself that must interact with core protein in some fashion that allows the cores to encapsulate that RNA. And then the question becomes, if you have a bunch of blue RNAs that encode blue core protein and a bunch of green RNAs that encode green core protein, and you co-infect cells with all those, okay, can the green RNA go in the blue cores and the blue cores go in the green, the other way around?
Yeah,
Okay.
blue, yeah.
And the answer is yes. That's trans. Okay.
Right.
So you can get the green RNA interacting with the blue cores and vice versa. Okay. The other question is the polymerase. Okay. If you have similar colors, I'm making this up as I go along, can a green polymerase be packaged with a blue RNA in the blue cores? All right. Well, not really the blue cores. We don't have to worry about that. But can the green polymerase be packaged with the blue RNA and vice versa? And the answer is no.
Yeah.
Okay. And I think there was evidence going into this that was the case. And that's cis. That means that you packaged along with the RNA from which you were translated.
Yeah, and another way that sort of, if you want to link some of this into Rich's description earlier, he described the virus first infecting the cell with that the gapped double-strand DNA genome, and mentioned that there was a polymerase there. So there's sort of the polymerase that came into the cell, and then there's also a new one that got made by translation
Yes.
off of the RNA. And so do we need that new one to do things that was encoded by the piece of RNA that we brought into the cell and that was made in the cell, or was that original one that came in good enough?
Right. So the reverse transcriptase is made on a messenger RNA, which they also call the pre-genome.
Right.
The question is, can that reverse transcriptase fall off and reverse transcribe a different pregenomic, or does it have to reverse transcribe the very one that it was made from?
Right.
That would be cis versus trans.
Right.
Right.
And going into this paper, the evidence already showed that Paul likes to dance with the one that brung him. Paul, the polymerase is going to polymerize the RNA from which it was translated. But we don't know why.
You took the title of this episode right off my head.
Paul's gonna, yes. Right.
So-
And the question of how that works is pretty important if we want better therapies against hepatitis B virus, because this is an essential part of the viral life cycle.
Yeah, and that's, they do figure it out, and that's why I like this paper, because
Yes.
I mean, the results are going in the textbook. This is a textbook paper where it's a basic new mechanism, and they're very clever because, as was pointed out, the genome is compact and the open reading frames overlap, so it's very hard to separate them. They figure out a nice way to do that here, and they use it to look at this question. So, what is it that they do? So, first of all, they say, can we uncouple the selection pressures on the core and the reverse transcriptase, which overlap, right? Is there some way to do that? So first they do a preliminary experiment where they insert termination codons in the different viral proteins, and then they ask, if we put RNA with those changes in it into a cell, will we get hepatitis B virus DNA? So that requires that the RNA is translated, that the reverse transcriptase be made, and that makes DNA from the RNA. So that's the readout, is Hep B virus DNA. So they introduce stop codons into the core protein coding sequence, the surface antigen, a protein called HBX, and then they measure. They don't want to put it into the reverse transcriptase because then you'll never get any DNA, right? And then they measure DNA after they put this RNA into cells by transfection.
And this is a case where it's totally legitimate to measure the viral DNA instead of measuring infectious virus. Because if
Right.
you measured infectious virus, then as soon as you messed with any of this, you wouldn't get any out.
Right.
Right, exactly right. So the surface antigen and the HBX functions are not needed for reverse transcriptase. You could put stop codons in those genes, doesn't matter. But the core is needed for reverse transcription, because to do reverse transcription, you have to assemble the RNA, the viral RNA, in a capsid composed of subunits of core. And so if you don't have core, you don't have that, and you don't get any reverse transcription. But basically now they have uncoupled the sequence requirements for Paul from the surface antigen and X in the regions where they overlap, and that's very cool. They then take another step where they say, can we make a library of sequence variants in core and Paul, particularly in the regions where they overlap, and separate the functions again by introducing those RNAs with the changes in them. So they use a method for introducing many changes in the viral RNA, and they make a pooled library of all different sequence variants. And so their logic is, they're very clever here. So if some of those RNAs make defective core proteins, they should still be encapsulated in trans by core proteins made by genomes that are okay, that are not mutated. So that's trans activity of the core protein. But if you make changes in the polymerase that are not functional, those are going to be removed from the population because they won't be reverse transcribed into DNA. So you will not see those when you look at the HBV DNA in your assay. So they do a very cool experiments where they say, okay, can we detect trans complementation versus cis preference for core and polymerase? So they mix wild type and mutant RNAs in different ratios. They put them into cells, and then they sequence the Hep B DNA that comes out of cells 2 days later. And the idea is, if the RT was absolutely cis-preferential, they would only get wild type DNA. But if there's no cis preference, which means it's absolute trans activity, we should get DNA with the same ratio of wild type and mutant as the input. What they found is that even when over 90% of the RNA that you put in encodes a catalytically dead RNA polymerase, and that's YMHH, those are 4 amino acid changes, nearly all of the viral RNAs, the pregenome RNAs, are converted to DNA, and they have wild type polymerase sequence, which means that the polymerase has a cis preference. It will only copy the RNA from which it is made. And so that result tells them they can now uncouple the requirements for core and Paul by using this method. Everybody got that? Does that make sense?
Yeah.
Mhm.
It's a little tricky.
Very well said. And this conceptually is really tricky. I have to sit with this for a while
Yeah.
to see it, and probably draw 40 different cartoons. They got a pretty good cartoon in the paper.
Yeah. Yeah, it's one of the reasons it's so unfortunate this is a paywalled paper, because it really does take reading it and thinking about it and looking at the figures And then for me, reading it again
Yeah.
and thinking about it some more and saying,
Yes, yes.
okay, now I
Mhm.
get it.
I'm still working on it, and I appreciate Vincent's description there because
Yes.
that helps.
So now they want to look at the requirements for core and polymerase in more detail. And to do this, I need to explain the current model for how this works. So again, in the viral DNA, the open reading frames for core protein and polymerase, they overlap. And so the current model is that ribosome, there's an open reading frame upstream of the core called C0. The model is that a ribosome translates this little open reading frame and it bypasses the methionine, the start codon of the core. Otherwise, it would make core. And so
Because
that
it's in a different
ribosome,
reading frame.
which is in a different reading frame, that ribosome then skips the core start site. It moves downstream. It reinitiates at a site downstream of C0. It then bypasses that one because the context, the sequence context, which we call a Kozak context, is not good, so it doesn't initiate there, and it moves downstream, and in doing so, it bypasses another methionine codon that's present in another overlapping reading frame, and then finally it moves downstream and it reinitiates at the polymerase open reading frame. So
So this is the leaky ribosome model.
right. So there are a bunch of
It
AUG
finds an
codons.
initiation site, but doesn't look so good. It passes
Moves
that.
on.
It doesn't look so good. It passes that. Finally finds one, says, yeah, this port is just right, and starts.
I really am amused by and this was news to me, amused by the idea that of these little peptides that
Yeah.
overlap
Yeah.
another initiation codon out of frame. So
Yep.
that would be a good initiation codon, even a leaky scanning model, but they're actually translating. So they go over that and they say, sorry, I'm busy. Right?
Yeah, but that's nice for you to say that, but we're supposed to dumb this down to a level of language that our listeners can grasp because you're anthropomorphizing like crazy right now.
Well, yes,
You're
we are, but that's
confusing me. You're
I
really
think that's
confusing me.
helpful.
So what I want to know, Al, is Vincent, when you did that nice little walk down through, and you mentioned this Kozak or Kozer,
Kozak.
Kozak.
the
Kozak.
Kozak, a zone which is unreadable for this particular polymerase. And then it goes past the methionine stop codon. And then it goes past another methionine. What gives it the right to do that? What
Uh,
molecular explanation can
a
you
lot of it.
supply that will tell you exactly where the skips should occur?
Well, it's not so much where the skip should occur. It's where do you have strong enough binding that you're going to stay around long enough for the next set of steps to
Do you
happen.
think this is van der Waals forces at work?
This goes back to the late 60s. Okay.
Well,
When
that's me.
protein synthesis was, when the genetic code was first being worked out, and it was realized that all proteins start with the initiation codon in all cases
Methionine.
is a methionine.
That's right.
And the question is, how come you pick out this methionine to start protein synthesis and not some other methionine?
Has
And
that
generally, yes, generally it turns out to be, well, there's two reasons depending what system you're in. In a eukaryotic system such as this, part of it is influenced by where relative to the 5' end the methionine is, because ribosomes initiate at the 5' end rather than internally. And they can bind to RNA and kind of slide without translating. And where they lock in depends on the sequence context of the methionine. There are other sequences, mostly upstream, that interact with RNA sequences in the ribosome and lock it in place and say, okay,
the
we're
sides get sort
starting
of
here.
held
Yes.
around that place. And it's not just the methionine. There's some holding on of the
Right.
side.
Start
too.
translating ahead. Use Burma shave, and then the ribosome.
And that's what we're
Jumps in.
calling a Kozak sequence, because some of the a lot of the information about that sequence was developed by Marilyn Kozak. And if it's a strong Kozak sequence, the dogma is that you lock in there and translate. If it's a weak Kozak sequence, you're likely to slide over it.
It has to do with the specific nucleotides at -1, -2, and -3. relative to the HEG and the +4. And that's all shown if you have the paper in the supplemental figure 2.
Ooh,
So-
supplemental figures.
Okay.
Yeah, we
Yeah.
did that.
So what they want to do next is use their mutagenesis scheme to look at what's needed to influence Paul reverse transcriptase translation. So they make a diverse library of mutants of these pre-genome RNAs. Every codon is changed to every possible amino acid in the core open reading frame. All right, and they put those RNAs into cells, and 2 days later they pull out the DNA and they sequence them, and they ask for every amino acid change, how does it appear in the viral DNA that we extracted compared to what we put in? That's called their fitness of each codon variant. So,
And this is the deep mutational scanning.
that's exactly right. And so
So it's deep because they're doing every possible change and they're scanning across this whole region.
the whole region
with
of
every
the
possible
core
change.
protein. And so if you introduce an amino acid, it appears as frequently in the viral DNA as it did in what you put in, then it has no fitness cost. But on the other hand, the other extreme, the change may completely eliminate the ability to make DNA, so it has a huge fitness cost. And they can calculate this by the percentage of the variant that ends up in the DNA. So what they find is really cool.
So this allows you to separate this otherwise obscure conjoining of core and pol because their reading frames overlap. So if you change a base and it messes up core, you could still get pol and you could still get reverse transcription. If you change a base and it messes up only pol, then you don't get reverse transcription.
And in fact, what they find is there is little fitness cost against mutations in the core reading frame upstream of the polymerase. So all that long part before the polymerase, you can change amino acids, and it's not a problem because core can come in trans, and so you can make some wild type core from other RNAs that may not be changed. But they do see signals of negative selection of fitness cost in the region of core that overlaps with the polymerase, and that's probably because you're making changes in the polymerase, and that doesn't give you a good amount of viral DNA. So that's a pretty straightforward result. But there is, in the core-only region, there are a few places where there is some fitness cost, okay? And one of them is methionine codons. There is a depletion of, whenever you change a codon to methionine throughout the core, it has a fitness cost on the production of viral DNA. And that is because if you introduce a methionine, now the ribosome is going to see it and it's going to start translating before it gets to the reverse transcriptase. So you'll have a decrease in the amount of DNA produced if you introduce a methionine. So that's why they say methionine codons are depleted throughout the core region.
You don't want to distract the polymerase.
You don't want to distract the polymerase. They also say there's an enrichment of in-frame termination codons upstream. So if you introduce a terminator, the ribosome will stop, and it allows them to then reinitiate and go to the polymerase start codon. So you're actually helping it. Of course, in the real world situation, it wouldn't help because there'd be no core. But experimentally, it shows you that if you help ribosomes get to the polymerase methionine, it's good. They also find, and this is important, 3 short vertical sites of negative selection in the core-only region. And these correspond to these places where we mentioned in that model that the ribosomes initiate in a short open reading frame, and then they resume. And so if you make a mutation, for example, that makes a really good Kozak sequence just by accident, then the ribosomes start to initiate there, and they never get to make polymerase. So it's totally consistent with this model of leaky scanning for getting to the polymerase. So I thought that was just so cool. It
Very
shows you
cool.
that
Yes.
it works really well. And all right, so next, they say, okay, we took all of the sequences of hepatitis B virus clinical isolates and we displayed them as a map with all the changes on them. And they say the sequences of the clinical isolates are nowhere near what we get from this deep mutational scanning, because the clinical isolates have to be infectious viruses, right?
Right.
And so any change that will screw up core that would still allow RT to be made in a clinical isolate would not exist. It would not infect people. So they say here, we can use our method to study codon preferences in a way that sequencing of clinical isolates simply cannot achieve. Okay, so now they focus on the Pol protein. They make a three-dimensional structure of it, and they find some interesting things that weren't known before, combined with their mutational map that we've just studied. Apparently,
And this is a predicted 3D structure.
It's a
They
predicted
didn't solve a
structure.
crystal structure. It's done with AlphaFold 2.
AlphaFold
AlphaFold
rears
2.
its beautiful head again.
Yes.
So
AlphaFold 2.
apparently there is a
Yeah, this is AlphaFold 2, which is actually documented and open source. AlphaFold 3, which was just published, is closed source to quite some controversy.
So they find a requirement for 4 cysteine residues in an N-terminal extension of the reverse transcriptase domain, which they find are likely to bind zinc. And actually, there are 2 of these. These are called zinc fingers, which are typically involved in structural positioning of proteins. And they find that this is conserved in other hepatitis B viruses of different species. And they basically think that this makes an extension that makes a loop in the RNA polymerase that actually corresponds to a loop in the surface protein that's targeted by antibodies, and that'll come back later. Now, in this AlphaFold 2 prediction, there is a C-terminus downstream of one of the domains of the reverse transcriptase that they found within that region, the mutagenesis didn't have much of an effect, except at 2 proline residues just before the termination codon for the reverse transcriptase. 2 proline residues. All right. Now, you may all know about 2 proline residues inserted into the spike protein to make the COVID vaccines. But here, they're doing something different. And proline is cool because based on its chemical structures, it's
more complicated to incorporate into proteins than other amino acids.
The key is that it will make ribosomes stall. So once ribosomes make 2 prolines, it will stall the ribosome. So they're thinking, well, maybe these 2 prolines stall the ribosome as it's making the end of the polymerase, the RT, right? Because these 2 prolines are just before the termination codon. And then that locks the reverse transcriptase onto the same RNA from which it was made, which is
the other thing arguing
which
for this.
Which is in cis.
Yes, which is in cis.
Exactly.
So the other thing arguing for this model is that this tail where these 2 prolines are is just kind of, it doesn't really have a distinctive structural motif to it. It's just kind of flopping there. And you can mutate the rest of it and it doesn't affect the polymerase. But these 2 prolines right there at the end would presumably slow down the ribosome as it's trying to, proline. I forget how to join those right. It's this other thing. It's not like anything else. And so the ribosome is stalled there trying to join these prolines, and that on the floppy tail would tether. It's just a very nice model in the pictures.
So they want to test this model. So they make some changes, and they make one of the changes that they change the prolines to alanines, right? And they've developed an assay where they translate this RNA in vitro in the presence of radioactive methionine. They can fractionate the proteins on a polyacrylamide gel, and they find a protein species migrating about 18,000 Daltons bigger than the predicted product, which would be consistent if it were still attached to a tRNA which has been stalled by which has been stabilized by a stalled ribosome. And they showed that when you replace the prolines with alanines, you don't get this species. So there's no stalling. The stalled species can be treated with RNase, which will digest the RNA, and it migrates at the correct size on a polymerase gel. So it's all consistent with the idea that these proline codons near the termination codon stall ribosomes, right?
So Dixon, you can forgive my anthropomorphizing here, I hope. But just to think about sort of the overall idea of what's going on here, we have the ribosome that is reading along the mRNA and is producing protein. And it's specifically producing the polymerase protein. And as it gets to the end of the RNA, it has produced a fair amount of amino acids to be the protein, and that ribosome gets stuck on working on the proteins, but it has already produced much of the N-terminus of the protein, and that N-terminus and that majority of the protein that has already been translated can fold and sort of become the reverse transcriptase. And because the ribosome is stalled, it's holding this newly made polymerase next to a piece of RNA. And so now, when that polymerase is fully made, it's going to be next to a piece of RNA and sort of be like, hey, look, I found a piece of RNA. Maybe
Might as
I
well
should
get
work
to
at
work.
this. Maybe I'll work on this one. Because it was held in place next to that RNA when the polymerase was being biosynthesized.
Insist.
Insist, yes.
Exactly right.
Hmm.
And they do one more experiment to show this. They make a mutated RNA, which has either wild type polymerase or that YMHH polymerase, which is catalytically inactive. Only 2% of the recovered DNA has the change in it. In other words, if the polymerase is catalytically inactive, it can't bind to that, can't remain bound to the RNA. But if you then change that RNA with the YMHH, you change the two prolines to two alanines, now you have 20% of the DNA recovered with the YMNHH, which shows that the diprolines enforce cis activity. Even if the polymerase is catalytically inactive, the diprolines could help it remain on the RNA. So
Right. So you replace those prolines with alanines, and now the polymerase can act in trans.
Yes. Right.
So the model then is that the polymerase translates the pre-genome, gets to these two prolines at the end of the reverse
this is the
transcriptase.
ribosome.
The ribosome is
Yeah.
translating the mRNA. It's making reverse transcriptase. It hits those two prolines, it stalls. And now, at the same time, core protein is being made. Core protein will package this pre-genome RNA along with the reverse transcriptase, and the reverse transcriptase copies, makes a DNA copy of that RNA, the same one that it was made from, because it was captured there at the 3' end during the packaging process. So, I mean, it just is a beautiful proof for that model.
Yeah.
And now the question now that they say, why does it work this way? Why is it cis-active? We don't know, but we would like to find out.
I think the hypothesis that really caught my attention and makes a lot of sense is this might be a final error check, because it will now you can only replicate a genome that has gotten successfully to the point of translating its own polymerase, which bodes well for it in the next cell.
Yeah, so if it's stopped anywhere along the way, it's never going to get to the polymerase.
It's never going to get there.
And they mentioned that this method could be used to study other viruses. And for poliovirus, people have thought for years that polymerase is the last protein to be translated from the RNA. And the model has always been it just sits there and copies that same RNA back. But nobody had any experimental data for it. But, you could use something like this. Well, I'm sorry, you can't work on polio anymore.
You can't work on poliovirus. You'd have to work on some other enterovirus. They do point out that they looked at other hepatinoviruses, and they see similar motifs among the orthohepatinoviruses. They don't see it among the more distant relatives, but they do see enriched prolines in the C termini in those viruses. So there may be a similar function going on there.
Now I can see someone who's going to ask us, but doesn't putting two prolines into spike cause this to happen? Right? But the answer is no, because there's no reverse transcription of the spike mRNA.
Right.
There's no copying of it. It's just made into protein, and that's the end of it.
It's a
But,
coincidental convergence of vocabulary.
Of two
Yes.
proleins.
It would
yes.
mean that when ribosomes are translating that mRNA, they would stall for a little while at those proleins potentially, but they do get past it eventually.
Yeah, and
Apparently they do. Yeah.
there's no other enzyme that's also trying to work on that RNA.
Right.
I mean,
And
what we
as
say
I
stall.
understand it,
ahead.
these are two adjacent proleins, right?
That's right.
Yes.
That's right.
Which is,
Two proleins.
that gums the works up significantly more than a single one.
Right. And the ribosome will get there. It's holding the polymerase, probably holding the polymerase there long enough for the polymerase to start work. And then the polymerase is going to replicate the whole genome, and it's already trapped in the core at that point, and so it doesn't matter.
So back to something that Rich was kind of musing about earlier was that when the pre-genome RNA gets out into the cytoplasm and it must somehow get recognized by the core, looking at this figure 6, I think it must be that it's getting recognized because of this packaging signal
Yeah.
epsilon.
That's my understanding.
Okay.
But there's another thing here that's interesting, and I'm on the edge again. The polymerase recognizes the RNA as well and binds to it. Okay, as part of this whole co-packaging thing. And there's in part A of that figure, they talk about an RNA packaging mutant that is somewhere in the must be a domain somewhere in the middle of the polymerase. And I'm thinking that you really need to. This thing is going to be folding as it's being translated. Okay, so you need to get down to the end and have the whole thing folded so that it can then, that domain can then interact with whatever RNA happens to be convenient, which, since it's paused at the end of the ribosome, is the RNA that's been being translated. What I don't know, I don't know whether there is actually sequence in the RNA itself that's important for the polymerase binding. There apparently is a polymerase domain. I'm sure this is known. I just don't know it.
Dixon, is this good with you? Dixon.
Yes, I'm saying yes. And all the time that you are all talking, I imagined Joe Pesci and Melissa Torme going through this on their my cousin Vinny defense. Now, are you sure that it's a cis translation, not a
Yes.
trans? I'm sure.
I just
Really?
watched that
And
movie.
how could you be so sure?
I
And then
just
she
watched that
says-
movie again the other day.
I watch it every time it comes on, and then or A Few Good Men, one of the other.
My cousin Vinny.
seats.
Only way you can handle the truth.
Give them this script, and they could allow the mental images to flow. I mean, you did anyway, Vincent. You did a good job. That's
Everybody
what I'm
did a
saying.
good job. We all kick in to do this.
Little
Yes,
sideline here. Marissa Tomei was interviewed on Finding Your Roots, a show that we all love, and she was raised in Brooklyn.
Yep.
Okay. So that accent is authentic. She said as she was growing up, her mother tried to cleanse her of the Brooklyn accent, saying that, this is going to do you no good down the road, because you're going to sound like a Brooklyn sort of, low life or whatever. Okay. And it got well.
She was a mechanic.
Part of what got her an Academy Award.
Daughter, right?
Yeah. What a beautiful movie.
Oh, absolutely.
All right.
What a beautiful story this was also.
Time for some picks of the week. Dixon, what do you have for us?
Okay. Well, very simple. I ran across this on, of all things, Yahoo News, which is one of my favorite news outlets, but mostly for the humor, not for the actual stories themselves. But this one appeared within that, and then it was a report from the New Scientist that they had isolated from a Neanderthal bone that was 50,000 years old. Upon doing electron microscopy on that sample, they discovered virus-like particles, and they look very, very, very real, right? I mean, the particles they almost look like little jewels. They're
I have
petrified viruses.
this behind a paywall. I have not been
Yep.
able to see the
Oh,
viruses.
no, but there are other outlets that'll handle that. Just
Okay,
type out 50,000-year-old virus, and you'll get it on your Google search.
I will do that.
I need to look at this because I want to know what they found. If they found it in the DNA or they saw particles, Dixon. Do
No,
you
particles.
remember?
It was particles. It was
Particles.
particles.
This is in the Neanderthal bones, right?
Yep.
Okay.
Oh, I see the pictures here. Traces of an
Yes.
adenovirus, herpes virus,
right.
and papillomavirus.
There you
So
go.
yeah, I see. Look at that. It's not just DNA.
It looks like one of your art things in back of you, Vincent, in
Yeah,
the
yeah.
incubator.
Well,
They're beautiful. They're absolutely gorgeous.
they look great.
They sparkle like jewels.
You
So
can get
this-
the bioarchive paper.
So Dixon, are they
See the full data.
infectious? That's the question.
Of course not. Of course not.
Why?
Of
Because they're
course
too
not.
old. They're mutated like
I
crazy,
think.
probably.
Well, no, they're.
I mean, because the
degraded.
particles look intact, right?
They do. They do. But those are proteins. Those are not the DNA
Yeah, I
or
understand.
the RNA.
Yeah, ancient DNA is heavily mutated because it's been under
Wow.
assault for many years, so it's unlikely to
Right.
be infectious.
Right.
But very interesting.
And
Cool.
Yeah.
so it pushes back by about 30,000 years the oldest virus recovered from nature.
Well, the oldest one actually recovered infectivity was Jean-Michel Claverie. He got some out of the Siberian permafrost, which
Yeah.
was about 40,000 years old. So this adds 10,000
And that's who
years.
did this work too, right?
Oh, no, that's better, though, because that was infectious.
Yeah, that was
This
infectious.
is just,
yeah.
it looks like a
This
virus.
is not the same group. No.
Oh, it's not the
Probably
same
a
group.
virus.
Okay.
No, it's from a group in Brazil.
Ah, okay.
The
Anyway.
investigator Marcello Briones says it may be possible to synthesize these viruses and infect modern human cells with them in the lab.
Oh, that
So
would be
it
exciting.
tells me that they're doing genome sequences.
Yeah.
Okay.
So I can imagine that
All right.
some people might not like that experiment, right?
That was my first reaction. Yeah.
But if you do the genome sequence and it isn't very different from a modern day adenovirus, then
Yeah,
I don't know.
well,
Well, we'll see what happens. We'll see if they do it.
how different was the influenza virus of 1918 versus the one we have today?
It's recognizable
Different
as
enough.
obviously you
Yeah.
only need one amino acid to make a big difference, right?
There you go. That's what I wanted you to say.
Who knows? Who
That's-
knows? Right. So they should just do it under good containment, right?
Right.
Yeah.
But very cool. Thank you, Dixon. Brianne, what do you have for us?
Welcome.
I have an article about a study done by a former Jeopardy player who is now a psychology PhD, who actually looked at the psychology of trivia knowledge and looked at memory systems in people who seemed to be particularly good at trivia, and found that many of the people who were very good at trivia had a unique way of encoding memory in their brain. So they had sort of a different memory system. Basically, there are two types of memory systems that we often think about, and her work showed that these two are kind of entwined with one another in Trivia Champions. And so people seemed to remember not just facts, but the context or the place or who they learned it from or something like that was always linked to the fact. And so I thought this was a really interesting to learn about. I didn't know a lot about these pieces of memory studies. I thought it was pretty cool that somebody took their experience on Jeopardy and made it into this. And I will admit that when I first read this article and read the description of the what the memory system was like and what the Jeopardy people's memory was like, I read it and I was like, really? Everyone doesn't do that? I don't understand. So I just thought it was a really cool way to learn about memory. And it also points out a little bit at the end that perhaps people could try to figure out ways to take advantage of it by trying to make sure that information is often taught with context and not just in a boring textbook or on flashcards or something, but I don't know, like in a podcast.
Yeah.
or like in a podcast, or like on a TV show.
Yeah.
That's right.
Kathy, what do you have for us?
So I have two things, and they're both kind of about fashion, fabrics, materials, and things that go into them. And the first one is about the bright future of intelligent fabrics. And both that article and the primary article are paywalled. So I'm sorry about that. But the gist of it is that there are, as you know, fabrics that clothes that can light up, but you need some kind of battery or something. But these workers have figured out a way to make chipless fabrics that can light up, and they do that by taking advantage of the human body and skin. And don't ask me too much about the electricity and that part of it, but they call these interactive fibers or eye fibers. And so you might wonder, why is this important? And first of all, they have a an application, which is that they make a wearable video game controller. They also have a smart carpet that could interact with other devices when it's walked on. But they also mentioned that it could be used in robots and robotic prostheses, which kind of leads into someone else's pick. I won't give that away. But anyway, it was just kind of cool that they have this whole thing worked out where these could facilitate extensive data gathering that you could use to train things or sense situations or things like that. So I thought that was cool.
Cool.
And then the second one, the main part of the article is not paywalled, and it's bacteria is the new black. And so it was known that you could have bacteria and coax them into making cellulose, but it was just this boring beige color. And so these people figured out that if they could combine it with genes that could make a black color in bacteria, then they could get something that was black. And so the bacteria is called Kamaguti Actor radicus. Comagadiubacter radicus. Anyway, I practiced it, but not recently. Anyway, it's the same bacteria that produces kombucha. And so anyway, what they were able to do is make these bacteria to make jet black cellulose textiles, which are kind of leathery. And so this would replace leather or pleather, which I hadn't heard of, which is plastic leather.
Plastic leather.
Mhm.
Yes.
And they fashioned it into a wallet and a shoe upper. And so I pasted that in from the primary article for you. And they also said that they figured out a way that they could put in regulators in such a way that when they exposed it to blue light, then they could get the cellulose pattern, the black pattern, in a pattern that would then look something like crocodile leather. So they're really working on how to get it so that it can be marketable, but it has kind of a long way to go. So anyway, I just thought it was funny that in these various science papers that I have been accumulating picks of the week, both of them were sort of about fashion and materials.
Cool.
Cool.
That's
Cool.
amazing. That's
So,
so cool.
Kathy, do you think that this material, if it responds to electrical impulses from the skin, and if there are some diseases which cause fluctuations in the electrical current in the skin, that this could be used as a diagnostic fabric, basically, for people suffering from unusual diseases like
Yeah, maybe.
eczema and stuff like that?
Or
Yeah,
heart
maybe.
disease, and it spells call 911 across your
Yeah.
chest.
Oh, that's right. Or
Right.
call me. Doing
Right.
anything tonight?
Yeah. Right.
Cool.
I don't know, but anyway, that was kind of neat.
Yeah.
Very
Or
cool.
vote Democratic. Vote
Oh,
Democratic.
and for the bacteria, they're also working on getting other colors besides black.
Oh
So.
yeah. Oh yeah.
Rich, what do you have for us?
So on several occasions, just a second, I went and booted that. On several occasions in the past, I had picks from a robotics company called Boston Dynamics. I've been kind of following these guys because they produce these just cool robots and cool YouTube videos about the robots. One of their primary robots that we've featured before is a dude called HD Atlas, who's a very humanoid robot that can do all kinds of stuff. And it turns out that after 11 years, they are retiring this robot. I think because the technology has evolved to the point where it makes more sense to start with a new platform. Okay. They've learned a lot from him, but they're not going to tweak him anymore.
They're going to
And so,
disassemble him and put him in a box like C-3PO in The Empire Strikes Back. And
Exactly.
so I got two links here. When they retired him, they made a sort of a tribute video. That's really pretty cool because it starts off with a bunch of failures, all right, of Atlas trying to do this and that and
Yeah, right.
stumbling and falling and stuff. And then it winds up with a bunch of successes, all right? So I found that tribute video and I've linked to that. There's also, I thought might be interesting, an article about Atlas's retirement that's riffs on the video, but also talks about some of the other robots that are being developed by other companies that will ultimately supersede this. So, for your amusement.
very cool.
It's a
Cool.
really great
Wow.
compilation video. I
Yeah.
had tears in my eyes, partly from laughter, but partly because I thought it was really poignant to see this thing failing and then eventually succeeding. It was really great.
Cool. Alan, what do you have for us?
So what I have, by the way, people should see the movie Chappie if they haven't already, if you want a robot story about that. But I picked this article. It's a good in-depth article by John Cohen, published in Science, about why it's not so straightforward to say, why don't we just vaccinate the cattle against H5N1? And you may have guessed that there was going to be some nuance here, and sure enough, developing a veterinary vaccine is not trivial. The USDA has put out a request for information from companies to find out, and from dairy farmers to find out what the parameters would be for developing something like this. There's, of course, the companies would be at some risk in developing a vaccine, not only because it might not work, but because even if it does work, this outbreak may have come to an end by the time they release it, and then they're out the development costs. There are other issues, like if you do vaccinate the cattle, then the milk may be subject to non-tariff trade barriers by other countries, where vaccination is often used as an excuse to block imports of cheap U.S. milk and protect farmers in other countries. So that's a concern. Of course, dairy farmers have their own concerns about the cost of this because it's a small margin business. And this article does a really nice job of just delving into all that and laying out what the issues are.
Nevertheless, are people working on it?
Yes, there are companies that do veterinary vaccines that are saying, yeah, we could. One of them, one of the veterinary vaccines for a different influenza. There's another platform. It's they use a baculovirus system, and they could just pop this in, and they say basically, yeah, we could start producing this right away. But then you get into all this other complexity, and so this all needs to be sorted out.
So
I
what do you have
love
to
the I
do?
love
Do you-
the picture on the top of this article.
The picture at the top with the vet giving the vaccine, just leaning over the cow.
So do you have to do clinical trials? No, it's not clinical.
They would
but-
have to do some kind of trials. So
Yeah.
that's one of the things people are hoping the USDA will answer, is whether they would accept a trial where you just do antibody levels. Because if you actually have to do a challenge trial with H5N1, which is a select agent, then you have to do a trial on cattle under BSL-3 conditions, and there are not a lot of places where you can do that, and they're not cheap.
It reminds me of the Nipah, the Hendra vaccine for horses, which was tested at a BSL-4 facility in Australia. They have a big enough facility they could test 50 horses in that facility.
Wow.
Can you
I'll be
imagine?
darned.
And
Well,
they had to do
Horse racing is a big deal down there.
they had to do necropsies at the end of the
Yep.
experiment.
Oh, yeah.
Not
Oh, yeah.
trivial, not trivial
Yeah,
stuff.
yeah,
But
yeah.
that's one place that can do it. But
Well, Notre Dame had a facility, I think they still do, where they raise germ-free horses.
Hmm.
So that's even more than a
wow,
BCL4.
cool. The only thing is that you can't really figure out side effects because the cows are not going to say
No,
anything, right?
no, that's right. That's right.
Yeah.
That's right.
Take him to the opera.
That's
Cool.
right.
All right, my
I think the biggest thing people would probably look at is milk production from those cows.
Yes.
You
Yes.
bet.
my pick is an article in Nature. Singapore Airlines turbulence: why climate change is making flights rougher. This is by Carissa Wong. I thought Alan would enjoy this.
Yes.
So there was a flight, a Singapore Air flight from London to Singapore on May 20th that suddenly dropped 1,800 meters and launched people and objects towards the cabin roof. One guy, 73-year-old man, was killed, 70 people injured, and they have a few photos of the cabin, which shows stuff falling out of the overheads. And this article is about how air turbulence is increasing as a consequence of climate change, and it's clear air turbulence. So it's not something that
Right.
you can. So, Alan,
Yep.
what do you see clouds and you assume there's turbulence? Is that how it is?
Right. There are different types of turbulence at different levels.
Mhm.
And when I flew, it was small airplanes that are down in the weather, and it was usually pretty easy to anticipate when there's going to be turbulence. Around here, we've got, what we call mountains. People out west would call them hills, but there are a lot of them. And when you get wind blowing over the hills, you always get turbulent airflow. You also get turbulence when heat is rising, like off a freshly plowed farm field. You can often anticipate it when you go outside and you look up and you see those puffy cumulus clouds. Those are actually a really good indicator in flight of where you want to climb up above in order to get into stable air, because those clouds are represent the rising air that's gotten up to a particular level. It forms a cloud when the air is not rising anymore. And so if you get above those clouds, it's usually nice and smooth. Now, in this case, so there's a lot of turbulence that's very predictable. And when you're in an airliner shortly after takeoff and it bounces around a little bit, or it's shortly before landing, it bounces around, that's that kind of thing. That's surface turbulence. This clear air turbulence is scary stuff for people who fly up in the flight levels. So this is up, 18,000 feet and above, and often like 30,000, 40,000 feet. You can just be cruising along, and you're not over the Rocky Mountains. You're not over anything that ought to be producing turbulence that high. But some weather systems, sometimes, depending on how much energy there is in the atmosphere, can generate these completely unanticipatable pockets of really, really severe turbulence. I know the Air Force has lost jets to this. It's bad, and you can see from the photos in this article. There's, as Vincent said, there's stuff falling from the ceiling, and the plane managed to make a safe landing, which is good. But this is something that can actually dismantle aircraft.
They had 20% spinal injuries on the flight.
Yeah.
20%. And that's
not
300 people.
Yeah, a lot of people.
I've flown that airline, but not from London.
Injured more than 70 people.
Yeah.
Yeah. So they cite some studies of clear air turbulence between 79 and 2020, and it's increased 55%, especially over the North Atlantic. So they say, so how do we detect this? They say they have a LiDAR that could detect it, but it's expensive. It's a big box, and it needs to be reduced in size. So you can see clear air 20 miles away. Although, Alan, 20 miles in a plane
20
going
miles, if you're cruising at 500 knots, it's going to come up on you pretty darn fast.
really
but
fast.
if you have some, a Betty warning or something that pull up, or roll right,
Yeah.
you could probably
That's right.
avoid a lot of
I
this.
just noticed in flying a lot that more frequently they say, flight attendants, take your jump seats. We never used to do that
That's right.
before. And
That's true. You're right.
seat
You're
belts
right.
on, way
Absolutely
more,
right.
sometimes 5-6 times on a
Yep.
6-hour flight. So that's kind of resonated. I've noticed it's more frequent. I'm
Yeah,
sure
you're right.
Yeah.
they're worried about it. I mean, can you imagine walking down to the bathroom and suddenly, bam, you get thrown up?
How about being in the bathroom?
Yeah.
So
Yeah.
I read that
Yeah.
one person, or that there was some shaking for a couple minutes before this happened, or maybe not even minutes. But anyway, this person that was being interviewed said that one person near him had time to put her seatbelt on, but no one else basically did.
Wow.
So
So that's why you should keep your seatbelt fastened all the
Keep
time.
your
right?
seatbelt on while you're
Yeah.
seated.
It's
Yes,
true.
like the sign says.
Very
Yeah.
true.
Anyway, scary stuff.
And of course,
Truly.
all the flying people are doing is making the climate change worse. So.
Yep.
Yeah, it's true.
That's right.
That is TWiV 1117. You can find the show notes at microbe.tv/TWiV. You can send questions, comments, pics to TWiV at microbe.tv. And if you like our work on TWiV and our other podcasts, please consider supporting us. Go to microbe.tv/contribute. Dixon de Pommier, trichinella.org, thelivingriver.org. Thank you, Dixon.
Good night, everybody, and have a wonderful weekend and enjoy the Memorial Day weekend. Vincent, load up on the pasta.
Yeah. Marathon loading. Okay.
Excellent. Excellent.
Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot. Definitely time to enjoy that awesome looking weekend out there.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.